Hello, ladies and gentlemen, and welcome to our discussion related to protein synthesis or gene expression. Now, before we get into this, I would like to encourage you to take a look at the Campbell website and spend some time looking at the animations and going through some of the interactive activities because they do a very nice job of sort of parsing out the concepts related to protein synthesis. So if you spend some time with that, it will help make sense of what we discuss here today. Now, the basic idea of this chapter is using the information in DNA to make proteins. Now, proteins do influence the production of our traits. You know, we are protein-based organisms. Much of our cellular structure is due to proteins, and enzymes run many of the chemical reactions of our body. So proteins are certainly primary in influencing our phenotypes. Now, we'll use bacteria as a model for much of what we discuss because there is this universal nature to the genetic code. These A's, T's, C's, and G's are found in virtually all forms of life, and the machinery used to take that code and produce proteins is also very similar between species and even domains. You know, archaea, bacteria, and eukaryotes all have this similar genetic structure. That allows us to take firefly genes and insert them into tobacco plants, or phosphorescent genes from jellyfish and put them into pigs that would look really cool at a roller skating rink. All right, now Gerard, basically all you need to know about his work is that he was able to demonstrate or postulate the idea that disease is a result of deficient genes which fail to produce functioning enzymes. Now, this idea was tested by Beadle and Tatum. Please make sure you are familiar with their experiments. It's highlighted in the text, and certainly there's opportunity to look at the box that goes over their results and conclusions. The basic finding of their work is the idea that a gene codes for the production of a particular enzyme. They developed what they called the one gene, one enzyme hypothesis. The idea is correct that a gene does produce an enzyme or a polypeptide, as we probably phrase it, but we need to expand that idea because certainly some genes don't code for enzymes. They can code for structural proteins like the keratin that fills up our skin cells. Also, some proteins are made of multiple polypeptides, like hemoglobin has polypeptide subunits that make it up. So again, one gene, one enzyme. We expand upon that idea, but the basic idea stands that genes produce specific products. Now, this is a nice summary of their results. The idea is these molds, or mold spores, can grow in what's called a minimal medium. It's like an agar or a jelly-like substance that has nutrients in it. So the wild type mold spores could grow in the minimal media. So they could basically assemble and produce their own amino acids and then create their own protein products. So the wild type could grow in minimal medium, as well as this minimal medium with specific amino acids added. But what they did was they zapped some of these with ultraviolet radiation, thereby damaging their DNA. So they tried to actually damage specific genes within these other mutant classes. And what they're able to find was that certain mutant classes would not grow unless provided a specific amino acid. What they're able to draw from that is that knocking out certain genes or damaging certain genes inhibits the production of specific enzymes. So as a result of damaging specific genes, they can inhibit the production of specific enzymes. This went and then provided empirical support to Gerard's predictions. So again, just familiarize yourself with that. All right, transcription translation. Be familiar with this idea in the central dogma that the information stored in DNA is used to produce messenger RNA, which then is decoded into the polypeptide sequence, or a sequence of amino acids that end up producing the protein. Now, this slide is basically about location. Transcription, we're making RNA, and it's done in the nucleus. In eukaryotic cells, we segregate the processes of transcription and translation. So transcription, again, occurs in the nucleus because that's where we find the DNA. Translation, on the other hand, occurs in the cytosol because, well, that's where the ribosomes and the amino acids that provide the raw material for the polypeptide are found. So these processes are segregated in eukaryotic organisms, whereas in prokaryotes they are not. All right, so getting into the differences between prokaryotes and eukaryotes. Prokaryotes, like bacteria, have no nucleus. Okay, they are prokaryotic, they were around before nucleus. So they can begin translating the messenger RNA into a polypeptide immediately after the messenger RNA is produced. They can even, before an entire segment of messenger RNA breaks loose during transcription, they can actually start translating that into a polypeptide sequence. Eukaryotes, on the other hand, do have this nuclear envelope that separates the processes of transcription and translation. So in eukaryotic organisms, we actually have fairly extensive processing of our RNA. Now, this initial RNA, the book will refer to it in different ways. They may call it the primary transcript, they may call it pre-mRNA, but the basic idea is it's the RNA that you first produce gets modified or edited in some way before you get this final messenger RNA that then goes out and is used to make the protein. So just to sort of highlight this, ignore all the words and just pay attention to the picture. You see that you take the large initial RNA and then it goes through some work here and then it gets spliced down or cut down into a smaller segment that then can travel out to the cytosol. All right. The whole purpose of this slide is just to sort of visually depict what we talked about in the previous slide. Here, transcription and translation can occur simultaneously without RNA editing. On the other hand, eukaryotic cells have transcription occur in the nucleus. We have this initial transcript or primary transcript or pre-RNA, whatever you want to call it. Notice how it has some material added to the end of the molecule, and see how it becomes shorter. It's been edited down or made shorter. That edited RNA then travels out to the cytosol where the information is decoded into a polypeptide. So editing occurs in eukaryotic cells but not in bacterial cells. Now, just to quickly hit the central dogma here, transcription is the process of converting DNA to RNA, again, in the nucleus of eukaryotes. And then translation takes that information from either the unedited messenger RNA or the edited messenger RNA and uses that to assemble amino acids into a protein. All right, now, this genetic code matters. DNA is the sequence of A's, T's, C's, and G's. It's billions of letters long in the individual species genome, potentially. So you have this code of A's, T's, C's, and G's. And in the same way that our alphabet with its letters has rules in which the letters are combined in different ways to produce different words with different meanings, this 4-base code of almost endless bases is read in a very specific way, 3 bases at a time, or 3 words at a time, if you want to think of it that way. Those 3-word segments are referred to as codons. And now, what is significant about a codon is the fact that each codon, each 3-base segment, specifies one particular amino acid. So remember, amino acids are important because amino acids link together to form the polypeptide. So there's one amino acid, and the overall structure is the polypeptide, or when that folds up, it can become a functioning protein. So the order of these codons specifies the order of the amino acids, which influences the 3-dimensional structure of the final product. All right, last little bit here. These codons, these three base segments, can be referred to as triplets. So, if you hear that, don't dismay. Just recognize that, codons are referring to these three nucleotide segments. And again, the key idea is each codon specifies a single amino acid. So, for instance, the codon or triplet AGT specifies the amino acid serine. Okay. Last slide here in this segment. With transcription, the whole idea is you use just one strand of the DNA. We have the double-stranded DNA, the strands are separated, and then this code in DNA is converted to the single-stranded messenger RNA. The thing to pay attention to here is that in RNA, here we have the RNA, in RNA, uracil replaces thymine. So rather than ATCG, in messenger RNA, we use AUCG. I just remember rather than at Centergrove, it's OW Centergrove.